me. Please join me in welcoming the incredible expert in the world of fungi, Dr. Amy Honan. Amy is a passionate mycologist whose curiosity about the hidden kingdom of fungi has taken her from the Rocky Mountains to the rainforests and beyond. She holds a PhD in fungal ecology and biogeography, and today she teaches mycology and fungal ecology at Oregon State University, where she inspires students with her infectious enthusiasm for the natural world. Her academic journey began with a Bachelor of Science in Biochemistry at Western Colorado University and continued through advanced research on fungal evolution and diversity. From the population genetics of root pathogens to the fascinating ecology of stuffed puffballs for her doctoral work. Amy's work not only explores the science of fungi, their systematics, evolution, and ecology, but also helps document and conserve fungal biodiversity across North America. She brings science to life, helping all of us see how fungi move, thrive, and shape ecosystems in mysterious and wonderful ways. We're so excited to have her here with us tonight to share her insights and enthusiasm. So please give a warm welcome to Dr. Amy Honan.
Yay.
Thanks so much for having me, and thank you, NAMA. These webinars over winter, I think, are so fantastic and keep us connected during our down season for fungi. So thanks, everybody, for taking time out of your busy lives to hang out and listen to talks about fungi. So today we're going to talk about how fungi get around, fungal dispersal mechanisms. And so I'm actually going to start with fungus-like organisms. So in your basic biology classes, you probably learned about protistans. So these are things that aren't in normal groups that we think of as normal groups. So we'll start out with a phylogeny. So this is an evolutionary tree, and this is an evolutionary tree of eukaryotes. So EU, that prefix means true, and karyo means nut. So these are everything that have a nucleus. And so I show this to try and get us to wrap our heads around all of the biodiversity that exists. And to orient ourselves to what we're looking at, there are some things that we know in this phylogenetic tree. So if we look in the upper left, we've got plants. So we all know what a plant is. And then in the bottom left, we have animals. And then over here, we have fungi. Recently, there's this other group of fungus-like organisms that we've been seeing. A lot of folks are taking these really cool pictures of things called slime molds. So I thought I'd start talking about slime molds. While they're not actually fungi, they're super cool. And historically, they were studied by mycologists because they are fungus-like. So this next little bit is for the taxonomy nerds. So if we think about the taxonomy of slime molds, they're in this group Amoebozoa. So you've probably heard of an amoeba. It's this blob-like thing that cruises around and kind of slurps up its food by squeezing around it and incorporating it into its body. So if we're thinking about slime molds, they're in this group, the Mycetozoa or the Eumycetozoa. And there's two main lineages that we think of for slime molds. We've got the Dictyostelium, which are our cellular slime molds, and the Myxogastria, which are the plasmodial or acellular slime molds. So here we have a life cycle diagram on the left, and we have a little video to accompany this. So we're going to start off with this life cycle stage of amoebas. So these cellular slime molds have one life cycle stage where they act like amoebas. They cruise around, they're eating bacteria and other things in the environment. And then at a certain point, they're going to get hungry and they're going to start signaling to each other to amass. So individual cells are coming together and they're forming this blob-like structure that's eventually going to start to differentiate, form these little knobs, and then they're going to slowly form this blob that's actually a slug-like creature. So here we have two different modes of dispersal. We went from an amoeba, that's an individual cell that can move around, then they all come together, individual cells amass, and they become this slug that can migrate. That slug, over time, is eventually going to also differentiate. That slug is going to develop a stalk or a stem, and some of the cells are going to cruise up to the top of this structure and form a spore-bearing structure. So these slime molds have this really crazy, intricate life cycle where they can live as an individual cell or they can come together and these individual cells will differentiate. So as this amoeba is cruising around, it could be eventually a stalk cell, or become a spore, or become any other part of this cellular slime mold. The other lineage we have are the Myxogastria or the plasmodial slime molds. And so you've all seen this structure, this blobby snot-like thing on a log. And so these are time-lapse images of this organism, and it is feeding. Again, it's eating bacteria and other things that are in the environment, and it's slowly pulsating through the environment. But this plasmodium is only one step in its life cycle. So if we look at that plasmodium, eventually it's going to produce a spore-like structure. So here we see that young sporangia developing. It's eventually going to make a stalk or stipe. And at the tip of that stalk or stipe, we're going to see the spores develop. So over time, this structure that's developing is going to differentiate into a different spore mass. And so the difference between these guys is that plasmodium is considered one cell with a bunch of nuclei. And so those spores are going to disperse, and those spores are going to germinate, and they're going to go down one of two pathways. Sometimes the spores are going to germinate into these amoeba-like structures. The amoeba, just like you would think of an amoeba, as this amorphous blob that's going to cruise around through the environment eating things. Or it can germinate into this flagellated life cycle stage where this thing with several flagella, one to several, will cruise through the environment. So at a certain point to complete the life cycle, whether it's an amoeba or this flagellated gamete, they're going to fuse at some point and become this diploid zygote. And so that zygote is what grows up to be the plasmodium. So in both of these organisms, we see a couple of different ways that they can disperse into the environment. So here's that zygote that's going to grow up to be a plasmodium. So if you're into these slime molds, there's a couple different ways that you can go about learning how to identify them. The most obvious one is to look at those spore-producing structures and the spore morphology, the capillacea, which are the little threads that hold the spores together. Then if you look at other life cycle stages, you're going to see if they're individual cells, if there's motility to them, if there is motility, are they amoeboid, do they have flagella? All those sorts of things are ways that you'll start to get to identifying these different slime molds. So that was our little jag off of kingdom fungi, but now let's dive into our fungi. And let's think about how fungi disperse. So just like we started with the life cycle of slime molds, let's think about the life cycle of fungi. So we all know what a mushroom is, and if we look on the gills of a mushroom, they're going to produce spores. And those spores are going to germinate into hyphae. And hyphae is kind of the business end of the fungi. So these hyphae are growing. They're these long filamentous strands. Those hyphae are exuding enzymes. Outside of the hyphae, the enzymes are digesting food, and then those hyphae slurp up all that nutrients so that the fungus can live. So fungi spend most of their life cycle in their hyphal stage. It's just when conditions are incredibly special that they're going to generate that mushroom that we all like to go out in the woods and find. So for our dispersal mechanisms, we're going to start thinking about hyphae, but we're also going to think about spores, so how spores disperse. And we're also going to think about the structures that produce spores, the sporocarps, things like mushrooms. So for hyphae, there's a couple of different adaptations of hyphae that I'll talk about. So one is mycelial cords. So what we see here are these long filamentous strands. They can be kind of fuzzy, and you probably have all seen this. If you peel off a piece of bark from a log, you see that sort of fan-like structure. And this is a way that fungi in their mycelial stage can disperse into different environments. And then we have much, much stronger than a mycelial cord are these rhizomorphs. So a rhizomorph is a mycelial cord that has a very thick skin on the outside. So this thick skin is melanized. And so just like melanin in humans is designed to protect us from UV radiation, The melanin that's produced on these is protective. So it protects from desiccation, UV, those sorts of things. It also confers a mechanical advantage. So it's very, very strong. So it allows these mycelial strands to push through the environment and move great distances while being protected. So here's an example of a rhizomorph, and we can see the reproductive structures, the mushrooms, coming off this rhizomorph. So it's analogous to a stolon. So if you think of a strawberry plant, those stolons are above ground roots, and it's enabling that strawberry to disperse into new habitats. And the same thing with the mushroom. So we have this rhizomorph, and in this case, it's actually reproducing via that rhizomorph. So we can move into new environments. And so this is an example of a marasmoid fungi that we would see in the tropics. We don't really get this kind of rhizomorphic diversity in North America or in more temperate climates. But if we go down to the tropics, we're going to have all kinds of different rhizomorphic diversity. You'll see these in trees, above ground structures. Sometimes they'll get caught, choke you out as you're cruising through the jungle. But what I want to focus on are these two pictures in the bottom. So in the picture on the left, what you can see is a bird's nest, and those white strands are rhizomorphs that are incorporated into the bird's nest. And that image on the right, you can see that bird is carrying around rhizomorphic material for the purpose of nest building. So a recent paper shows us that at least 176 bird species use fungal material in their nests. So a cool example of this fungal material in nests are with these orioles. And so on the left, you can see that oriole that's hanging out in its nest. It's this big drapey kind of nest. And there's a cross section of this nest on the right-hand side. And so what happens is these birds spend a lot of time looking for rhizomorphic material or fungal material. So they're skipping over plant material in search of fungal material. So they're spending more time doing this. And if you're an animal and you're searching for a certain material, that's money. So metabolic energy. So it's costing you something to go search for this material. So it better give you a benefit if you're going to spend metabolic energy looking for something. And in fact, it does. So these fungal materials are stronger than plant materials. They have higher tensile strength. So if you have a stronger material, you can hold more. So that means that they can have larger offspring and they can have more offspring. So that's contributing to the fitness of this bird and the reproductive success of the bird. In addition, this fungal material is water repellent. So if you're decreasing your water load, you're decreasing your pathogen. And so you're protecting your offspring or anything that's in that nest. And in fact, some of these rhizomorphs actually produce antibacterials. So there's some marasmoid fungi that are antibacterial, further decreasing your pathogen load. So all of these things contribute to increased fitness and increased reproductive success. So using fungal material is advantageous to these birds. Another good example of rhizomorph and the importance in fungal biology is Armillaria. So here we have Armillaria stoyi and the rhizomorphs of Armillaria stoyi, and this is our humongous fungus. So it's considered one of the biggest organisms by mass, area, and volume. So the humongous fungus in Oregon is between 2,400 and 8,600 years old. It encompasses almost 2,400 acres, which is about 37 square miles, and it's estimated to be about 35,000 tons. And so this fungus is one individual. It's reproducing via rhizomorphs. And so this one individual takes up all that space by reproducing asexually through rhizomorphic dispersal. I do want to give a disclaimer because in 2022, there's a seagrass, I'm not going to say this correct because it's not a fungus, Posidonia australis, which reproduces via rhizomes, which is analogous to a rhizomorph. And it apparently is the largest organism by square size, and it's 49,000 acres. So the humongous fungus got bumped out by the seagrass, but it's still one of the world's largest organisms. Another hyphal adaptation that we have are sclerotia. So this is kind of like a pocket of mycelia with a melanized outer coat. And so this is a structure that enables a fungus to be dormant for a while and then germinate when conditions are more advantageous for the fungus to grow. And so we know that at least 85 different genera and 20 orders of ascomycetes and basidiomycetes produce sclerotia, and these structures have evolved independently at least 14 times. So an example of a fungus that produces sclerotia is Claviceps purpurea. So what we have here, this guy right here is sclerotia. So what happened was the sclerotia fell to the ground, went underground, overwintered, and then in the spring, these spore-bearing structures germinated from this sclerotia. So what's going to happen is these spores are going to disperse via wind, usually, and they're going to land on the inflorescence of a grass. And what they're going to do is they're going to infect the ovary of this grass. During the growing season, they're going to produce these little structures in those ovaries that are sticky. And so the fly or some sort of flying insect is going to come. The canidia are sticky, so they're going to stick to that fly, and then that fly is going to go into the next inflorescence. So during the growing season, it's making structures that can reinfect different plants. So it's getting dispersal mechanism that way. And then as fall approaches, they're going to produce these hardened sclerotia. These sclerotia are going to fall out of the plant and fall down to the ground and overwinter. And the reason that I use this as the example is because Claviceps purpurea produces this compound, which I'm sure you all recognize as lysergic acid. So lysergic acid is LSD. And so if we think about these plants that it's infecting wheat and rye. And what do we make out of wheat and rye? Delicious things like bread. And so you can imagine what would happen if you ate bread that has been infected by Claviceps purpurea that has lysergic acid in it. You're going to feel a little bit funky. And so this is one of the theories that was of why the Salem witch trials occurred. So if you're not familiar with the Salem witch trials, it was a series of hearings in the late 1600s where many people were accused of witchcraft in Massachusetts. So over 200 people were accused of witchcraft and 30 people were found guilty. And these people were all executed. So 19 people were hanged and one poor fella was actually pressed. What they did was they took him between two pieces of wood and they put a bunch of weight on him. And so that's how he suffered. So there's many different theories that about the witchcraft that was occurring during the Salem witch trials. And this is just one theory, is that folks were eating bread that was infected with claviceps. So they were basically just tripping on acid. All right, so let's move on to spore and sporocarp dispersal. So here is a phylogenetic tree of our fungi. And so the fungi that most of us are familiar with are the Basidiomycota. So those are things that have gills, those are gilled fungi. And then the Ascomycota, so things that are like cup fungi or morels. We also have a lot of other diversity in this group in fungi that we don't see and that aren't immediately obvious. So in your introductory biology textbooks, you probably saw a phylogenetic tree like this. We have quotation marks around some of these lineages because we now know that they're much more diverse than our textbooks taught us. This is a little bit more realistic of a phylogeny, and I just show this again because I like to have folks understand the amount of biodiversity that we have in the world. And so this is kind of flipped from the phylogeny that I showed in the previous slide. And so these guys down here, we kind of think of, or you might hear them referred to as early diverging lineages. And so we think of evolution on Earth. We moved from an aquatic environment to a terrestrial environment over geologic timescales. So a lot of these early diverging lineages are adapted to water, whereas these we call Dicaria up here have more terrestrial adaptations. So the first group that we're going to talk about is the Chytridiomycota. So the Chytridiomycota are more adapted to aquatic environments. So they have these sporangia, and in these sporangia, they produce spores that are adapted to an aquatic environment. So they have flagella, so they are able to swim. And so here we see the sporangium maturing and those flagellated spores swimming through an aqueous environment. And the reason I bring up Chytridium mycota is because we have this really important fungus in this group called Chytridium dendropatidius, which is really fun to say, but we prefer to say BD because it's much, much easier to get off the tongue. So what we're looking at is that sporangia structure embedded into this here. And what we're looking at over here is actually the skin of a frog. And so this BD, this fungus, causes this disease called chytridiomycosis. So I'm going to warn you, the next slide might be a little bit graphic. Here we have the disease in all its glory. So on the left-hand side, what we see is that sporangium coming through the skin of a frog, and this is what happens in extreme cases of chytridiomycosis. And so if you're an amphibian, you have very small lungs because you are able to breathe through your skin. So as soon as we start decimating that organ on these frogs, they lose their ability to do gas exchange, and it's very devastating. So this fungus was thought to originate on the Korean Peninsula, and it's been distributed throughout the world. And so what was going on was there's frog trade for a couple of reasons. Some of it was pet trade. One of the biggest things that was going on is that these frogs produce a compound that's used in pregnancy testing. So these frogs got transported all over the world for lab testing, for pregnancy testing, and for pets. And so we see this. What happened was the frogs that had BD, they co-evolved with it in the Korean or in that Asian area. But when those frogs brought that BD into new habitats, the native populations were not adapted to this fungus. And so this fungus ran wild and started attacking other organisms that hadn't co-evolved and didn't have the immunity to this fungus. And so what we're seeing is a mass destruction of amphibian populations due to the spread of this fungus. So at least 2,800 amphibian species are threatened. An estimated 40% of amphibian species are threatened globally, and at least 90 species have gone extinct through this process of moving a non-native species into new areas. And so if we look at the genetics of this BD, we can see that there's little genetic signatures that differ throughout the world. And so let's focus on North America. So in North America, we have the vast majority of this is this GPL lineage, but we have this little sliver that's from Brazil. So what's going on there? And the reason that I'm interested in this is because I live in Colorado, and we have these beautiful alpine lakes that I like to visit. And in those alpine lakes, we have this boreal toad that was threatened even before the introduction of BD. Now, with the introduction of BD, anywhere that the frog has been exposed to BD, there's complete extinction of frog populations in those areas. And if we look at the genetics of BD in Colorado in these alpine lakes, it's the genetics from Brazil. So this is still a question that's ongoing. How did the Brazilian lineage get into these alpine areas? And that might be a question that we're never able to answer. And then this is kind of a little bit of a dogleg, but I think it's very interesting. So if we look at the phylogeny of the chytrids, they're in this kind of blue region. This is the phylogeny of all the chytrid fungi. And at the tips of all the branches are what they eat. So we can see all the different ecologies of what chytrids eat. And if we zoom in on where this BD fungus is, it's on this branch. And if we look at the tip of this branch, these chytrids are eating vertebrates. Sister to that are saprotrophs, so things that break down dead organic material. So if you think of that, that's a pretty crazy jump in ecology to go from breaking down wood or plant matter to attacking a vertebrate. And then if we look at the other groups in there, these are things that attack green algae. So chytrids are just a crazy, crazy group. And we're seeing some mass destruction from the movement of this one chytrid, this BD. And so I just want to touch a base on a few definitions. So when we talk about species ranges, where a species exists, we can think about their native range. That's where they occurred historically, where they evolved, and even a little bit smaller pocket of a native species is an endemic species. So an endemic species is restricted to a certain location, so a much smaller area. And we can contrast this with non-native species. So these are species that are now in an area in which they did not historically occur. And there's many different levels of non-native species. We have introduced species, we have established species, and we have invasive species. So an introduced species is a species that is living outside of its historic range. And so introduced species are 99.9% of the time introduced through anthropogenic means, meaning a human moved it there. So an introduced species can become established if it becomes a self-sustaining population without human intervention, meaning we're not planting it, we're not making it reproduce. It can do it on its own without us doing anything. An invasive species is an established species that's causing harm. So an invasive species is going to displace other species, it's going to disrupt ecosystems. So that BD fungus is an example of an invasive species. We introduced it, it became established, and it's very clearly causing harm. And so we see this over and over again with introduced species. We have Dutch elm disease, which is caused by Ophiostoma ulmi and closely related fungi. So this is believed to be native to Asia. It was introduced to North America in the 1930s on a shipment of infected elm logs. And so it rapidly spread throughout North America. By the 1980s, it had killed an estimated 75 to 80% of the 77 million elm trees in North America. And by today, it's estimated that about 95% of elm trees have been decimated by Dutch elm disease. Another example is white nose syndrome or Pseudogymnoascus destructans. And so kind of the same thing, it's thought that this was introduced to a cave by some equipment. So some cavers went in and they had this fungus on their clothes or shoes. So it was introduced in Eastern North America in 2006. And since then, it's killed about 7 million bats across North America, and it continues to creep across North America. Recently, it's come into Colorado and Arizona, and we can watch the spread of this. Another one is this Favelatia, which is just this beautiful, spectacular, poroid little mushroom, but it's pushing out native colonizers. It's super aggressive in wood. And so Favelatia is one of those really easy ones to track because it's so bright, it's so orange, you can see it. But you could imagine if you go back to our fungal life cycle and you think about that hyphal stage where fungi are living, it's very difficult to detect when a fungus is introduced, when it becomes established, and when it becomes invasive, because we can't see those hyphal interactions that occur within wood or underground. And so I see a lot of folks moving fungi around, like we have these new products that people are introducing. People are advertising wag bags that decompose human feces. So they're saying, bring it up into the alpine so you don't have to carry your feces and you can just bury it with this little fungus pellet. People are cultivating fungi, right? The golden oyster is a great example of a cultivated fungus that's becoming invasive. I've seen recent documentaries where people are doing restoration work and they're using fungi, but it's not necessarily native species. So just thinking about how we move stuff around, because we don't know, we don't have enough information about how fungi interact in nature. And it's very, very difficult for us to understand if a species if we introduce a species, is it going to go down that pike of being established and then invasive? All right, so let's move on to some more fun stuff. So let's move into the Mucoromycota and the Mucoromycotina. So this is one of my favorite fungi, the Pilobolus. And so what we're looking at here is this little translucent stem has an eyeball on the top of it. And so what we're looking at here is a cartoon of this eyeball. And these little lines, these indicate light. And so the light is hitting this structure at the base of the eyeball. And what the fungus is going to do, it's going to orient itself so the light is equally hitting each side of the fungus at the base of this eyeball. And the reason for this is at the top of this is the spore mass. And so it knows if light is hitting it evenly here, this spore mass is unobstructed and it can shoot off and go and disperse, and it's not going to get blocked by anything. And so if we look at a video of this, we've built up so much hydrostatic pressure in that eyeball that it shoots this spore mass off with incredible velocity. And so here we have a comparison. This top is going to be a rifle. This bottom is going to be a shotgun. And in the middle, we have the pilobolus. So go ahead and place your bets on who's going to win this race. And we're going to see which wins. So there goes the rifle, the shotgun, and then the pilobolus. So you can very clearly see that that pilobolus has the fastest acceleration, and it's over 3 times faster than a rifle and a shotgun. So we have the biggest fungus, with the exception of the seagrass. Fungus is the biggest, the largest, the most mass, and then we now have the fastest. And so the reason that they're doing this is they're trying to disperse from their substrate. So here we have the pilobolus shooting its spore off. It's going to shoot to the grass. This grass is going to get ingested by an herbivore, and then it's going to come out in the waste of the herbivore. And so most organisms, despite some of our dogs perhaps, don't like to eat their own feces. So they need to get away from their feces so they can continue to finish their life cycle. And there's one more little part to the story. So if we look on top of some of these pilobolus, we see this little guy, which is a roundworm. So if you've ever had to deworm an animal or a pet, this is what you're doing, is you're getting rid of these roundworms. So these mammalian pathogens are hitching a ride on pilobolus. And so we've got these really awesome dung fungi researchers like Sigrid and Alfred who are doing really cool slow motion photography. So I'm going to challenge you guys to get a video of this roundworm coming on, like on that spore and shooting off. And I have a theory that it's got a cowboy hat and a lasso and it's going wee through the whole sky as it's getting shot off. So we'll see if anybody can get a video of that at some point. And so pilobolus is not the only mushroom or fungus that exhibits phototropism. So there's other fungi in this group. So these types of fungi you can grow. So if you get a little dung chamber, just get some herbivore dung. You can see these kind of fungi grow and you can play around with them. You can hear the spore mechanism shoot off. They're really fun to look at and grow. And then everybody you live with will be really excited that you have little pockets of dung in your house growing funky little things. All right, so the next group of fungi that we're going to talk about are some insect pathogenic fungi, so the Entomoptera And so you've probably seen different videos. Here we have an ant that's been infected by insect pathogenic fungi. And so I'm just going to back that up a second. So what's happened is this ant has climbed up, and then right there, what's happening is the fungus is causing the mandibles to grip onto the substrate. And so the fungus has made this ant, glued it into its substrate. And the ant is also high up, all because it's trying to raise up its spore-producing structure. So we can see that spore-producing structure come out of the back of the ant, and then the spores are going to be distributed throughout the environment. And what's really slick is somehow they generally know where the ants are, particularly with ants, because the ants put a pheromone out and tell the other ants where to go. So this ant that's infected will climb up above that line of ants that's cruising back and forth to the nest. So this fungus has a better chance of dropping its spores on other insects to be able to continue its life cycle. And so we can see this in other insects too. So here's a fly that's been infected with an entomopathogenic fungi, and the fungus has taken away this fly's ability to fly. We're messing with the fly. It's not flying anywhere, but it is showing the summiting thing. So this fly is climbing up. It's doing something it normally wouldn't do. It's not flying. And then its mouthpieces are also manipulated by the fungus. So we see the mandibles come out of the fly and actually get glued to the substrate, just like we saw with the ant getting glued to the substrate. So the fungus has taken over all the musculature of the insect. Here's the next thing that happens. The fungus is moving the wings of this insect out of the way. And so in a second, we're going to see why these wings need to move. Now we can see the fungus is coming out of the abdomen of this insect, and it's shooting its spores off. So by moving the wings out of the way, the fungus is ensuring that it's getting dispersed into new environments. And so this structure that's being produced is called blastoconidia. And so here we see that forcible discharge of those spores, like we've seen a couple of times now. We build up hydrostatic pressure and we shoot the spores off. And these spores are so smart. Here we see this structure in the back is a wing. And so here's the original conidia that landed. And this fungus recognizes that it's not on the substrate that it wants to be on. So it's producing another forcible discharge because it can't do anything with the wing. It needs to get to the insect. So it's making another spore. And as you might imagine, this is crazy. They're all the different chemicals and proteins and genetics that go into this coordinated effort to make sure that these entomopathogenic fungi can overtake an insect, make the insect do what it wants to do, and disperse its spores. So as if that wasn't enough, they also manipulate the reproductive behavior. So here what's happened is this infected fly has attracted a non-infected fly to mate with it. And so it's mating with it, so it's getting its spores. And then if that wasn't enough, it produced a glue. So this poor guy that tried to mate with another fly to make more flies is actually just a vehicle for dragging around this carcass of spores to continue this mechanism of dispersal. And so we see this also in the cycads. So what we're seeing here is right here, the whole abdomen of this poor little cycad has fallen off, and we see the spore mass in this abdomen of the cycad. So this cycad essentially has no reproductive value other than for the fungus. And I think this is a great example of scientific communication. So a lot of this work came out of Matt Cassin's lab, and so they coined this the flying salt shakers of death. And so these spores are flying through the environment. This cycad is still alive somehow, even though half of its body's gone. And it's producing pheromones, again, to convince other cycads to come mate with it. And it is surviving on a fungal diet of amphetamines and psilocybin so that it can continue to live and spread its spores. So these fungi have come up with all these mechanisms with which they can reproduce and ensure their dispersal and ongoing survival. All right, so let's move into fungi that we know. We've got Ascomycota and Basidiomycota. So again, the Basidiomycota are basidiomycetes. They reproduce via basidium, basidiospores, and these are our guild fungi, things like boletes, that sort of stuff. And then Ascomycota, those are our cup fungi and things like morels. And so we touched on the basidiomycete life cycle earlier. If we look at the gills, they have these structures called basidia. On the tips of the basidia, we have basidia spores. And if we look at the basidia, there's all kinds of diversity of basidial shapes. So we have things like the rusts and smuts here. We have jelly fungi that make these tuning fork shaped ones. Then we have things like this is what we're going to see on our guild fungi. So these basidia make basidia spores, and those spores are forcibly discharged. And we call that ballistospory. So if we come to the tips of a basidia, we have this little structure called a basidia. And what you're going to see is that spore is asymmetrical. So at the tip of the bottom or base of the spore, we get this little droplet called the Buller's drop. And so what's going to happen is eventually that Buller's drop is going to get large enough and it's going to roll down this flattened surface of the spore. And this area is hydrophobic and this tip is hydrophilic. And there's some sugars involved, but this momentum is going to force the spore to break off as that water droplet rolls down. So here, if we increase the surface tension, you can see how that water droplet got sucked up that spore and shot it off. So all these basidia produce these forcible discharges. And so fungi are amazing engineers and physicists. If we're thinking about forcibly discharging between two gills, two lamellae, that spore can only go a certain distance and it has to drop. Otherwise, it's going to hit the other spore. So the spore has to be the right size and the right shape, and the size of the Buller's drop also has to be the right size to create the perfect amount of velocity so that spore doesn't hit the other gill edge and then it just drops down. And so I'm not going to talk too much about spores and spore dispersal. Dr. Elsa Balinga has a tremendous talk that she does on spore dispersal. So hopefully you guys get a chance to see that someday. But I will talk about how spores create condensation. So what we see here is this is after the spore has dispersed, after the ballistospory. So that Buller's drop is gone. These spores actually have the ability to generate more water. So at the base of that spore, what we're seeing in these images is another water droplet is being formed. And so what's going on here is they used a scanning electron microscope that does environmental samples. And in different levels of humidity, that spore can actually produce condensation. And we can see this with many different species of spores. So here again, we see condensation forming on the spores. And so if we look at a cartoon of this, the spore shoots off. It's got no water, but then eventually in the atmosphere, it can start causing condensation of moisture in the atmosphere to where it's actually producing a water droplet. So let's take a second and just talk about the water cycle real quick and the significance of the spores generating condensation. So this is a simplified diagram of a water cycle. So the sun is going to hit some body of water like the ocean. It's going to cause evaporation. So condensation is going to go, or water vapor is going to go into the atmosphere. Different pressure, temperature is going to cause that water vapor to condense into a cloud. And if conditions are right and there's enough water vapor in the right conditions, the condensation is going to fall back to Earth as precipitation, perhaps rain, perhaps snow. And then eventually, hopefully, that water is going to return to the body of water, and that cycle is going to continue. So if we zoom into a cloud, for precipitation to happen, we need this formation of an ice crystal. And so for an ice crystal to form, we need a nucleus. We need something that that water ice can form on. And so we call this an ice nucleus or an ice nucleation particle, INP. And so if we have this ice nucleus, different liquid droplets can attach to it. They can freeze, and over time, this frozen body is going to get larger, and if it gets big enough, it's going to fall as precipitation back to Earth. And so the key is you need that nucleus for precipitation to happen. And so folks have been looking at this and looking at these INP or these nucleation particles. And recently it's been discovered that, particularly in the Arctic, that most of these ice nucleation particles are actually fungal spores. So fungal spores are responsible for precipitation, largely for precipitation, particularly in the Arctic. And so they analyzed those particles and found that they're actually basidiomycetes, but very little is known about them. We don't know which species are contributing the most to it. And as we know, all fungal species do crazy different things. So there's a lot of work going on this. But if we summarize that, we have spores that enter the atmosphere. They cause condensation of the water vapor, which creates rain, which increases humidity, which creates favorable growth conditions for most fungi. So we see this cycle happening and happening. And so we see this positive feedback loop. So more fungi are causing more precipitation, which are generating more fungi. So that's pretty interesting and a huge reason why we should be thinking about fungal conservation, because without fungi, we might see changes in our water cycles. So we talked about ballistospori and basidiomycetes, but we also have ballistospori and ascomycetes, so the cup fungi. So you've all probably seen a cup fungus, and if you breathe hot air into that cup fungus, you can cause the forcible discharge of the asci, the spores that are in that ascomycota. So if we look at a cup fungus and we take a cross section, instead of basidia, we're going to see asci, these little soldiers that are all lined up. And within each of these finger-like structures, we're going to see the spores. So if we start to zoom in on those structures, here we have an ascus with many spores, and it's going to disperse its spores. And it's going to do it so quickly that it's very hard to see. And so here's a different structure or a different type of ascus. It's got a little lid on it. So when it shoots its spores off, we're going to see this little operculum left. So if you're somebody who is interested in studying ascomycetes, these are the kind of the structures that are very important, these little features. Does it have an operculum? Does it have a staining reaction? And so what we're seeing here is Ascobolus, and they have these really cool pigmented spores. So within those dung fungal chambers that I know you guys have all already started, you can grow this fungus too on herbivore dung. So another one, and you can see this macroscopically. You can actually see these guys shoot their spores off. So we'll just quickly switch back to basidiomycetes. So I want to talk about a little bit about the evolution of the shapes that we see in basidiomycetes. We can all picture a gilled mushroom, but there's many, many different structures that these basidiomycetes produce. So one trajectory of evolution in basidiomycetes is basically increasing the surface area of the spore-bearing structure. So if we think about some of these types of fungi, these clavulina type fungi, they're just these little fingers. And then the spore-bearing surface is wrapped around each of these tips of the branches. Over evolutionary time, these single branches are going to branch into multiple branches and turn into more of a coralloid fungus. And so what we see is a difference or an increase in surface area. Once again, we have another increase in surface area where the mushroom produces a cap and then the gills drop down from there. So we have an increase in surface area. And then if we throw in some cross walls, we have some poroid fungi. So an increase in surface area. So that is one trajectory of fungal evolution. Another one is becoming enclosed. And so if we take a gilled mushroom and instead of opening the cap and showing our gills, if we keep them enclosed, we have what's called a secotioid, or some folks say a secotioid structure. And so inside this, you can still see the mottling. So there's still gills in here. You can still see basidia, and the spores are produced in this structure. And then over evolutionary time, you can't even make the gills out anymore. All the spores are produced inside this very, very enclosed structure. If we were to cut that in half, there's no semblance of gills left. They're just these long strands called capillacea, and the spores are produced there. And so this process of gastromycetation, or producing this enclosed structure, has happened at least 123 individual times over the evolution of fungi. So every lineage of fungi is going, almost every, is going to have this enclosed form, and it's happened in ascomycetes and basidiomycetes. So all these different lineages are going to have this enclosed form of fungi. So if we think about this process, we have some fungi that have become enclosed and they're epigeous. So epi means upon and geo means earth. So these are puffballs that are above the ground. And so if we think about this, we have this structure, we have basidia, but if there's ballistospory, it's pointless, right? If we shoot a spore off and it's enclosed, it's not going to go anywhere. So these guys rely on some perturbation. So some environmental factor, an animal walking by and brushing up against it, a raindrop, something like that, that's going to shoot the spores off. And we have these other ingenious little mechanisms. So in Astreus, we actually have a layer of tissue on those rays that absorbs water. It's hydrophanous. It causes those rays to expand and then eventually reflex, and it's going to lift this spore mass up and off the ground. And this is happening while it's raining. So the fungus is smart enough to know, all right, the rain's coming. This is my time to shine. I'm going to lift up and make sure that my spores get dispersed during this rainstorm. And the other big transition is a change in shape. So I mentioned in those ballistospores, we have those asymmetrical spores so that that buller's drop can run down it. In the gasteroid fungi, we have round spores because we no longer have forcible discharge. What's also interesting is these round spores can also cause condensation of water, but they do it in a little bit of a different way. It doesn't come at one side of that spore like we saw in the asymmetrical spores. They kind of make this halo. And so again, the ratio of different types of basidiomycetes that are forming those ice nucleation particles, we're not really quite sure, but definitely an area of interest of folks like looking at what kind of spores we're seeing in the atmosphere that are causing rain. So besides epigeous fungi that are enclosed, we have hypogeous fungi. So hypo, under, geo, earth. So these are our truffles, right? These are enclosed fungi that grow under the earth. So again, we're seeing these ballistospores that are asymmetric, changing to these round spores, and they're called statismospores, meaning they're static. They aren't forcibly discharged. And then we see this process, whoops, of going underground. So these are truffles, and they have all these adaptations to enable spore dispersal. So if you're underground, a mechanical disruption is not going to get you very far. So like a raindrop isn't going to help. So we have these different adaptations in these truffle fungi, these underground ones. And one of the big ones is spore morphology, right? So we have these hugely pigmented, often very spiny. And as soon as I said truffle, most of you guys had a thought in your head and you probably smelled something, right? So we know that these truffles are making sense, that are attracting organisms to dig them up and eat them. And then we have this adaptation that the spores can pass through the animal's digestive tract to enable them to survive. They're going to come out in the waste and they're going to be dispersed via animals. And so that brings us to some animal fungal dispersal mechanisms. One of the first ones I like to talk about is fungus farming. So insects were the first farmers, and we see multiple different types of organisms actually farming fungi. So we have things like ants, termites, and beetles, all farming fungus. So in each of these instances, a different insect brings a fungal colony to a new environment, so dispersing it into a new environment, and then each of these insects are responsible for tending to these different gardens, and these different gardens feed these insects young. So they each have different mechanisms, different specializations. We see all kinds of really significant coevolution that occurs in these different systems. And we could talk for days and days about all of these. You're getting the sense that I'm talking really fast because I have a lot of different stories and I'm just touching a little bit on each of them. And we see mycophagy in all kinds of different animals. So back to the birds again, we have this one study that these folks were in the tropics and they were looking at these truffles and they were seeing all these weird scratches and peck marks and things, and they were wondering what was going on. So they spent some time looking and they saw birds visiting these truffles. So as any good scientist would do, they went and looked at the bird poop. And within the bird poop, they found spores, and they also ran DNA analysis and were able to confirm that birds were eating these truffles. And what better dispersal mechanism than a bird? They fly, and they're enabling the dispersal of these fungi. A lot of them were ectomycorrhizal, and enabling the dispersal of these truffle-like fungi to another ectomycorrhizal or another tree area to form these ectomycorrhizal relationships, which promote the reforestation of different areas. And then mammals, just like us, right? So tons of mammals eat fungi. At least 508 mammal species eat fungi. And again, determining what species of animal or what species of fungi the animals are eating. Folks are looking at dung. They're analyzing the spores that are in the dung and the DNA that occurs in dung. Um, and it's not just birds and mammals. Reptiles like skinks and turtles eat fungi. And then slugs, we've all probably seen a slug eating fungus. And what's really interesting is, again, with the dung of slugs was analyzed, and they found that the spores that pass through a slug, found in dung are still viable. So all these things are contributing to the dispersal of fungi. We have all these animals moving fungi around. But is it really just the bacteria that are dictating the movement of all this fungus? So if we look at this, here we have a scanning electron micrograph of fungal hyphae in bacteria. So if we zoom in, we see these long fungal filaments, and associated with these fungal filaments are bacteria. So bacteria are prokaryotes. We talked about eukaryotes earlier. Pro means before, and karyo, again, nut. So these things they lack a nucleus. And so these organisms evolved before eukaryotic organisms. And so let's talk about the interactions between bacteria and fungi and see if we can figure out maybe the bacteria are dictating some of this movement of or dispersal mechanisms in fungi. So we're going to come back to the slime molds because they have some interesting interactions with bacteria. So if we come back to this structure, the slug. So remember I said these guys are cruising around and eating bacteria. And so if we look within these slugs, they have these cells called sentinel cells. And so as the fungus is moving, it's sloughing off some of these sentinel cells in that slime trail that slug is leaving behind. And so if you think back to The Matrix, your sci-fi movie, right? So you have the human resistance that's fighting against the Matrix. So we've got Trinity and Neo fighting against the Matrix, but the Matrix has these sentinel cells that are going after the human resistance to try and kill the human resistance. And so that's the same thing that's happening within these slugs. We have these sentinel cells, and they're going against the bacteria. So the bacteria are being eaten by these sentinel cells, and then some of these sentinel cells are getting sloughed off in those trails and continuing to fight the bacteria that are in the slug's vicinity. But not all the bacteria that are contained in those sentinel cells are being eaten. So in this image on the right-hand side, we have some of those slime mold cells, and they're empty, so they don't have bacteria. But on the right-hand side, we have what are called farmer slime mold cells, and those pink little or neon colored structures are bacteria. So when folks first started discovering this, they called the farmer cells because they have this bacteria. And so these slime molds were thought to be taking these bacteria and seeding their next area. So they had a food source. And so what they're now finding out is that these bacteria are actually conferring what is thought to be some fitness advantages to the slime mold. So what we're going to see on the right-hand side is these are the slime molds that have bacteria, and you're going to see that the stem is higher compared to the slime molds that lack bacteria, and that spore-bearing structure is larger. So these bacteria are conferring an advantage to these slime molds. In theory, by being able, with a taller stipe, you're able to disperse farther and you're making more spores. And those spores contain bacteria. So it is fulfilling the reproductive needs of the bacteria because the bacteria is getting dispersed into new environments. So here we have an example of an interaction between a slime mold and bacteria that seems to confer advantage to both participants, even though the slime mold can sometimes consume the bacteria. The bacteria is ensuring its ongoing survival by associating with these slime molds. And we see this other interactions with bacteria and fungi. So here on the left-hand side, we've got this Penibacillus vortex, and on the right-hand side, we have Aspergillus. So Penibacillus is a bacteria that forms these colonies, and it lives in the rhizosphere, which is the area around plant roots. And on the other side, we have Aspergillus fumigatus, which is a widespread saprotroph. You have this fungus in your compost heaps. So on the left-hand side, we have a still, and on the right-hand side, we have a little video. And so this blob mass is the bacteria, and all those dark spots are the conidia from the Aspergillus. And so what's happening is this bacteria is actually transporting the spores of the Aspergillus. And we can see this in this rotating colony. Same thing. So this bacteria makes this rotating vortex and it moves the fungal spores around. So here we have this really interesting dispersal mechanism of the spores that are utilizing the bacteria, or maybe the bacteria is utilizing the conidia, and they're both dispersing through this interaction. Here we see another dispersal mechanism that the bacteria are utilizing the fungi. So we have these long filamentous strands, and around that filamentous strand, we sort of see that halo. So that is the extracellular matrix of the fungus, and that's where all those enzymes are being exuded by the fungus. And here we see the bacteria are swimming up and down that aqueous environment. And the bacteria are flagellated. They need some sort of aqueous environment to transport. So here we have this interesting interaction where the bacteria are utilizing this fungal hyphae to disperse themselves. And so this is thought to be the precursor, or where bacteria started associating with fungi. And now the bacteria, we are starting to see that they exist in many different fungal strains. So within the hyphae of many different fungi, there's bacteria that live inside the fungus, and it's thought that this is how it got started. And we're just starting to peel back the layers of all the interactions of bacteria and fungus. And one place that folks have really been studying is how bacteria interact with truffles. So again, I say truffle, you imagine that delicious smell that walks by you in a restaurant. Again, that smell is developed for dispersal. Many other different things that are truffles produce many volatiles for many different reasons. So we have these sulfur volatiles that attract animals and insects, but they also produce alcohols for signaling. They produce esters that attract small insects and soil animals, and then they also produce some other volatiles that are for their immune system, for protection or deterring other things. And so what folks are starting to realize now that a lot of those scents are actually made by the bacteria that live inside the truffles. So if we look at the bacteria are inside these truffles, so we see all these images are different truffles. The green structures are the fungal structures, and then everything that's labeled B and is red is bacteria. So within these truffles, there's a whole microbiome that exists, and there's succession. So the bacterial colonies or the bacterial populations change over time within these truffle communities. And if you look at these volatiles that exist within the truffle community, some of the scents that are made, they are made by the fungus. But a lot of the scents that are made by the bacteria. And the other layer that happens is the fungi are sometimes producing the precursor to the volatile, and the bacteria comes in and does some sort of enzymatic rearrangement of that chemical to make the volatile that we all want to eat. All right, so then we have this crazy fungus here, Guaianocastor necrorhizis. So this is a fungus that Kathy Aim and Terry Henkel found in Guyana many years ago. And so here is a cross section of this fungus, and we can see it maturing over time. So this is kind of a subhypogeous gastroid fungus. So it remains enclosed throughout its maturity, and it's kind of lives right at the intersection of earth and ground. So you can see it sticking out. And so what they noticed about this is that it stays enclosed throughout maturity. So one of their first questions was, what is this? So they did a phylogeny, and they found out that it is sister to armillaria. So we talked about armillaria earlier, right? It's the humongous fungus, and it reproduces via rhizome. So there's a lot of asexual reproduction that happens. So they were able to generate those rhizomorphs in culture of this Guaianocastor. So from there, they were wondering how does this thing reproduce? Is it asexual like a lot of armillaria, or is it sexually reproducing? And so they figured out that it is sexually reproducing. But it didn't really make sense because there was no point in its maturity that they could see that the spores were exposed. So if the spores were enclosed, how are they getting out and reproducing out in nature? So within the fruiting body, within these structures, there was no oxygen. But they also noticed that there was increase in nitrogen with maturity. And so there's not many things that can increase nitrogen. One of the most prominent organisms that can increase nitrogen are nitrogen fixing bacteria. So what they noticed is over time, the bacteria, nitrogen fixing bacteria, increased in these mushrooms over time. So what was going on is lots of things need nitrogen to live, so that can be attractive to different things. And in fact, what they figured out was that it was attracting termites. And so not only was the nitrogen attracting termites, there's also pheromones that attract the termites. So the termites come in, they eat this sticky gleba, the spore mass, and the spores stick to this termite. And then this termite cruises off into the woods and deposits the spores onto the wood, and then that fungus is able to create its life cycle or complete its life cycle. So I've touched on a little bit of the ecosystem services that fungi provide. We see them in all plant tissues. We see them in the roots, in the leaves, in the stems, in flower, in seeds. They have different interactions with animals, from mutualistic to commensal to pathogenic. They're in aquatic environments, they're in marine environments. Within the roots of plants, they're incredibly important for mycorrhizal associations. Most plants need an association with fungus. And then they're the world's most important recyclers. So if we didn't have fungi, basically none of our organic material would have ever been recycled, and we'd live in a much different environment. And despite this overwhelming importance of fungi on the ecosystem, we know veritably little about fungal biodiversity. So here we have an image, this was developed by Q, that shows the relative biodiversity by size of each of these images. And so the numbers in black are the described species in each group, and the numbers in green are the estimated number of species in each group. So if we start over on the left with that tiny, tiny little vertebrate animal, about 74,000 species have been described of about 80,000 known species. So that means we know about 92% of the animals that exist on the planet. We know about 88% of the plants. We know about 18% of the invertebrates. And we only know about between 2 and 6% of the fungi that exist on the planet. So there's no way that we can work towards understanding all these crazy stories of dispersal, their ecosystem services, or even work to conserving if we don't know the biodiversity. So we absolutely need to work on documenting biodiversity so that we can work towards our conservation efforts. So we have a couple of mechanisms that we've been working on to get to this conservation idea. So one thing that the Fungal Diversity Survey has started is the Rare Fungi Challenges. So we have one in the Northeast, we have one in the Southeast, we have one on the West Coast, and we have one in the Rocky Mountains. So over the past 5 or 6 years, each of these surveys have been able to engage the public in documenting fungal biodiversity. So you'll see that each of these challenges were started in different years, and each of them have had different numbers of species. They each have 20. And so each of these challenges have been able to document some of this fungal biodiversity. And gathering this data is invaluable in our ability to really understand fungal abundance versus rarity so that we can use these data to argue for conservation of different fungal species in these different geographic areas. Without this data, we don't have a way to make the argument that these fungi are actually rare or abundant. And so there's many people that are in charge of documenting these data, and they do it for free on their own time. So in the Southeast, we've got Alicia Milliken, who's working there. In the Northeast, we've got Hannah Huber. I'm sorry, Kamal, I have not met you and I could not find a picture, but Kamal is the coordinator for the West Coast Challenge. And then Mila Essam is the coordinator for the Rocky Mountain Challenge. So we owe a huge debt of gratitude to these folks who are volunteering their time to document, work with the public in documenting these. Another avenue that we've been doing is building a pipeline to utilize all our databases to really count fungi and understand the abundance. And so Scott Bates was part of the development of the Mycology Collections portal. So this is the database that all natural history collections utilize to document fungi. So like the Chicago Field Museum and the Denver Botanic Gardens, you can go into the Mycology Collection portal and see their collections. So what he was able to do is pull in records from iNaturalist and MycoPortal and basically count all the fungi that exist in these different areas. And the goal is that we can figure out the abundance and rarity of each of our fungal species in different geographic regions to be able to rank them on NatureServe. And NatureServe is what's utilized by most natural heritage programs to monitor and track rare species. So plants and animals, insects, these things are ranked on NatureServe, and the natural heritage programs in each state utilize this data to come up with monitoring programs. And so what we're finding was when we go into each of these, basically all the species that we find are ranked S1, the most critical, which we know is not necessarily true. So we really need to continue to work on documenting our biodiversity so we can get these numbers to improve our numbers, improve our data. So when we go to rank these, we know we're using best data. So what a lot of folks have been doing, we've been going around to different events through different clubs and working with the community on documenting fungi, working with natural history collections so that we can better understand fungal biodiversity so we can really work towards these conservation efforts. And so hopefully you guys got to see Rick Vandepoel's talk on fungal conservation. If you missed it, it's available. The webinar is available on the NAMA website. And so we have all these amazing people that are working towards fungal conservation in North America. So we have Dr. Jesse Allen, who just started as the mycologist for NatureServe. We've got Hannah Huber, who's the state mycologist in Pennsylvania. We have Jackie Brand, who just started at the Fungal Diversity Survey, and then we've got Nora Dunkirk, who's the state mycologist in Oregon. So all these folks are really working towards conservation efforts throughout North America. And so we really have to give a huge debt of gratitude to everybody who's working towards that. I know I went a little bit long. I'm very sorry, but thank you for your time, and I'm happy to answer any questions.
Amy, thank you so much. That was absolutely amazing. Round of applause, everyone, for Amy. No, honestly, that was a great presentation. There was so much dynamic things going on. It was not your standard PowerPoint presentation. I loved it. We do have a handful of questions that have come in. If anyone has any additional questions that they would like, feel free to use the Q&A function. Hopefully we'll get to them. We'll try to go quickly through the questions so that we can get to what we can in the time that we have. All right, so we got two first off on rhizomorphs. So the first one are the rhizomorphs. Are they used, the ones used in the bird's nest that you were talking about at the beginning? Would they be completely dead when they're taken by the birds? And if they are alive or dead, would they still be producing the like antibacterial chemicals and whatnot?
That's a great question. So generally speaking, they're thought to continue to be alive. They're like they have potential, right? So they can be dormant, but they have the potential to germinate. And so a lot of the times they're going to still be they're going to have those chemical properties that are conferring advantages. When they are moist, they could continue to germinate into the some of them are going to produce those sporocarps, but some of them are not. Some of them are just going to be rhizomorphs.
So the second question on the rhizomorphs is you said that there weren't many that grow out in the northern areas of the world. I assume kind of in our areas, you say down in the tropics that are a little more plentiful. What's the reason behind that? Surely they can evolve to deal with our weather, you know?
Because the tropics are nuts. When we think of a mushroom, like all bets are off in the tropics. What they like that Guiana castor, right? That's an armillaria. It's very closely related to an armillaria. If you were to pick that mushroom up, there's no way in the world you'd think, oh, that's an armillaria, right? It's enclosed, it's highly nitrogenated, it has this thick outer layer, it's got this sticky gleba. So all the adaptations that we see and everything that we consider to be a fungus is so different when we go down there.
But we do have some rhizomorphs in the north.
We do, like armillaria produces rhizomorphs, but the way that fungal reproduction happens, another big thing is the amount of nutrients in tropical soils is counterintuitive that it's very, very low. And so if you are able to be aerial, you can get to different nutrient levels and avoid kind of a dearth of area. And there's sometimes more carbon available in canopies. And if you're elevated and producing a spore bearing structure, when you're elevated, you have the potential to disperse farther.
Fair enough. Sclerotia, are they more legume-like in structures or are they similar to truffles?
So a legume is a pea, right? So that's a reproductive structure. So sexual recombination has made a seed. And same with a truffle, right? We have spores in the seed. So it is, oh, my brain went blank. What's an asexual reproduction structure of a plant? That's like, right? So like reproduction hasn't happened. Pollen hasn't met an ovary to make the sclerotia, right? It's just a mass of hyphae. So a tumbleweed, right? Something that isn't making a seed. Does that answer that question?
I suppose.
So no, it's not a legume and it's not a truffle. Superficially, it's going to look like a truffle.
All right, we'll take that. Take
Yeah.
that as an answer. Could chytrid propagules be transferred from Brazil to Colorado on the feet of or in the guts of migratory birds?
That that's the going hypothesis. How would you ever test that? How could you generate data to support that? That's the that's a big question.
Yeah, pluck the birds from the sky.
Yeah.
Yeah. All right. So the another bird question here. So do the birds eating the truffles that we had mentioned? Are they digging up those truffles, or are they just eating truffles that are kind of left over or sitting around on the ground?
It they were digging them up.
That's
Yeah.
interesting.
So the those truffles are going to get dispersed by any organism they can convince to dig them up and move them around.
Those ones must be relatively shallow, or they bigger birds.
I think both. Yeah, so like, if you've ever gone out with the truffle dogs, right? Some of there's stratification, right? So there's some truffles that are quite close to the surface of the soil, and there's some that are deeper down. Um, they don't seem to be too deep.
Okay. Um, any knowledge on how long spores can persist in the environment, viable or not?
Yeah, so s- um there's all the work on. Oh my gosh, you guys, my brain just went dead again. But if the folks who are doing the coring and looking at ancient soils and they're looking at pollen in those to try and figure out ancient dispersal of plants, there are fungal spores in there. There's just not that many people who are trained to look for fungal spores in those ancient soil cores. And so they can exist for a long time. I didn't really talk about spore morphologies at all, but you can all think of a dark pigmented mushroom spore and a white pigmented mushroom spore. So those dark pigmented spores are melanized. So anything that's dark is going to persist in the environment much longer than any of our non-pigmented hyaline, not pigmented white type of spores. And so like the dark pigmented ones can maintain viability for much longer. It's very species specific. Those truffle spores, right, that are super ornamented and pigmented, they're going to have much longer viability than something like a Lepiota spore that's thin-walled, that's clear, that doesn't have any pigmentation.
The word apparently we were looking for was palynologist.
What's
The
that?
person, palynologist is apparently the person who studies the fossilized microscopic items. So that was the word that was thrown out in the chat. Um, so a couple more questions on the chytrid there. So one was just a anecdotal story, I suppose, with a question attached, which was recently found a northwestern salamander, seemingly unhealthy with lesions on the underside. Um, Submitted to iNaturalist, no action was taken. It has since died. Is this something that should be checked for chytrid Moses?
There is an so I didn't go into emerging pathogens, but there is an emerging chytrid on salamanders, and it is in North America. And I don't know who is currently looking at it. Based on where you are, you might contact the Department of Wildlife in your state. Um, I would imagine that the Department of Wildlife in every state is tracking the chytrid that's on salamander, and it's called like salamander vensis or something like that. I'm blanking on the name.
Interesting. Okay. And one of the follow-up kind of questions about that was how to reduce the spread of that. Is there something that we can do as individuals to reduce the spread?
Yeah, so they're aquatic. So if you are going into wet environments, you want to just like anything, bleach your clothing, your shoes. Like if you've been to Hawaii and you do like go into ohia areas to prevent the spread of rapid ohia death, right? You're going to scrub all the mud off your things and just be very careful. Like, and I probably don't do it well enough, but like if we're collecting fungi, being very diligent about our collecting equipment, our backpacks, our knives, clippers, all that sort of stuff. making sure that when we move into different environments, we're not transporting anything that could be transported. And especially if we're in an area where we know that there's something invasive like a chytrid fungus or something that's causing disease, being very diligent about not transporting them.
That's a good call. And I think that's true for most native species and stuff like that as well, right? And seeds and spores and everything. While we're talking about kind of spreading these spores and stuff around, would inoculating a bird feeder with rare native fungal spores be a valid way to spread and proliferate rare fungi?
Yeah.
Yeah.
Because clearly that's how we do it. Um, it depends on the biology of the fungus, right? So, um, do they disperse via spores? Are you going to get them onto the substrate that they need? Why are they rare? Or is it just a rare mushroom, but it's actually common in the habitat, meaning like the hyphae are there, we just don't see the mushrooms produced? Do they only produce a mushroom like a cycad? They only come every 7 years. But yeah, let's put the rare fungi in the birds and get the birds to do our work for us. I think that's a great idea.
How do glomerulus mycota disperse?
Oh, geez. So glomeromycota are arbuscular mycorrhizae. Um, and so these are really interesting. We don't see them. 90% of plants are arbuscular mycorrhizal. So they exist in the soil and you never see a reproductive structure. So for a very long time, we didn't think that there's any sexual reproduction in them. We now know that there is. And so in my lifetime, we went from, I don't know, having maybe 60 described. I don't study these, so this number is probably not right. Last I looked, there was probably 250 described species. And it's not really known how they disperse. Or I guess I don't know much about their dispersal because they don't produce above ground structures. So they just are kind of cruising through the environment underground, and you don't see them very many places. Like, so if you'd expect them to be above ground and get dispersed, you would think that you would find them in different plant structures, and they don't seem to be turning up in other environmental samples.
Um, how do spores get up into the atmosphere to form that precipitation?
Oh, so I mentioned the ballistospore, right? So usually what happens, like if you saw the cup fungus, right, you blow into the cup fungus, so you change the humidity. When you blow into it, they get put up into the wind column, and some of them actually generate their own wind dispersal mechanisms. So basically, they just get caught up in the wind. There's been a lot of studies on the distance of how far fungi disperse, and some of them don't disperse very far. So we have examples of like ectomycorrhizal fungi on the forest edge. It does not seem like spores go very far, but there are folks that have sampled in the atmosphere and have been able to find tons of spores up there.
Um, growing mushrooms by saving the spores from the wild. Is that something that is possible for all kinds of mushrooms?
Yeah, I think what most people do is they do spore prints, right? So they take a spore print and then utilize the spores from the spore print to germinate fungi. I don't really grow that many fungi. Um,
Yeah. Um, so there was one question here, clarification on armillaria producing asexually, and whether uh, being called that armillaria does not produce through asexual spores. Is there a
right.
misunderstanding there?
Yeah, they reproduce asexually via rhizomorphs.
Okay. Basidiospores assisted cloud nucleation appears to be a self-fulfilling cycle for the fungus, whereby the ensuing rain ensures spore disposal. There wasn't a question there, apparently. So, Another one was also that there was even newer emerging chytrids on snakes in the Northeast. So all
Yeah, I
fun
think
stuff
we're
like
going to
that.
continue to see the mutation of vertebrate attacking chytrids. So a good example of emerging infectious diseases, which is a great career path for any students out there.
It's too bad. It's a great career path.
Job security.
Yeah, there you go. That's all the questions we have. I don't think we have any more in there. Just a reminder, people, that Amy did make reference to a conservation talk. I did post it into the chat. So if anyone wants to go over to YouTube and check it out, I'll post it again. It's on the NAMA YouTube channel. And this presentation will also be on the YouTube channel. Thank you, Amy, for that. I appreciate it. And yeah, I think that's about it. Oh, I got one more question in here. Can fungus survive in space?
Um, some spores can survive a vacuum, but I don't know, like, can a mushroom, can hyphae? Depends on the structures.
There we go. We'll leave it at that then. Amy, thank you so much. Everyone, thank you for joining us this evening. It was fantastic. Amy, you are perfect. We so much appreciate you having come and provided us with this amazing information. And we hope to see you again on the NAMA webinars.
Awesome. Thanks so much. Thanks to everybody. Thanks to NAMA and Trent and Brooke and all the folks that keep NAMA running. We appreciate you. Thanks.
All right.
Bye.
Thank you, everyone. Take care.